We're at the bioanalysis Zoom, and we have a few experts in bioanalysis. Today, we're going to talk about advancing bioanalytical method development and validation for small molecules in a changing regulatory landscape. This is Jin Wang. I'm a senior director of bioanalysis at the Kinetics Pharmaceutical.
I'm Jennifer Zimmer, Laboratory Director at Alturas Analytics.
GSC Chen, Director of Small Molecule Bioanalysis in ICH M10.
Raghu Ramanathan, QPS, Newark, Delaware, Executive Director there, handling biotransformation and GLP workflows.
Nong Yuan, Director at Biogen.
That's great. Yeah, thank you everyone for your time. And today we're going to have a few topics. First, we're going to talk about the importance of a bioanalytical validation. We know bioanalytical method development and the validation is the foundation for our business and at the pharmaceutical companies or at the CRO bioanalytical labs. And that's what we do. And we have panels and working in the pharma and in the CROs as well. Some of us have been working at the both locations. So you get the experience from both places. Hopefully we can exchange our current thinkings, what the current status for the regulatory requirement and how you do your method development and validation at your different places. We can change notes and hopefully we can all learn something and today. Is that sounds good?
It sounds good.
Yeah, right, great. So I think first question is really why bioanalytical method is important. And Raghu, could you share some of your thoughts?
Yeah, definitely. Bioanalysis, it spans from early discovery through post-marketing, and it is to ensure that we are bringing safe drugs to the patients. And some of the bioanalysis methods, starting with early ANMI screening into safety tox studies and going into first-in-human studies, and then in patient studies. So it is a wide opportunity for us to contribute and make sure that we are bringing safe drugs to patients. And that's why bioanalysis is very important. And we've been really fortunate that a lot of guidance documents also allow us to make sure we have the scientifically sound methods coming in and we're delivering to make sure we have safe drugs on the market.
Yeah, that's great. Could you comment on what's the requirement from regulatory perspective from FDA and from EMA and right?
The regulatory perspective is to make sure that we have a method that's scientifically valid and scientifically sound method. And most of the regulatory guidance establish that you have all the requirements, specificity, sensitivity, to make sure that we achieve the needed sensitivity to detect these drugs and metabolites in samples like plasma or tissue samples and whatnot.
That's great. Thank you, Raghu. And JC, I know you're managing a quite big team to method development and the sample analysis. Could you give us some idea or share your thoughts on what's the typical method development process in terms of to select the technology and LCMS or other platforms and how to do the optimization in terms of what parameters you're going to look at? Could you share some thoughts?
Yes, so for small molecule bioanalysis method development, so normally when we get the project, we need to understand the analyte and also the matrix. So to understand the metabolism, to understand the protein binding, and first we need to initiate how to do sample preparation and extraction. So either liquid extraction, protein precipitation, or solid phase extraction. So after that, so we need to select the analytical techniques. So normally we do LCMS, MS, so MRM method. So you can also depend on molecules and the matrix. You can choose a higher resolution mass spec to detection or LC, UV, or liquid binding assays. So then we need after that we need to understand how to optimize the parameter. So we need to know the sensitivity, we need to know the specificity, and also matrix fact. So then we can go to the method parameters to go through all the method parameters, LQ, ULQ, like linearity and accuracy per scene to confirm we can build a robust method for future validation.
That's great to see. Yeah, Jennifer, could you share some of your perspective in your company and what validation parameters you're looking into, and also what regulatory guidance we're following or is relevant to our work?
Yeah, so I think our sponsors are always looking globally, right? So we have to not only look at in the past we had to look at the FDA guidance, and then there was European guidance, and so we were really lucky to have some of the M10 guidance, the ICH guidance, that is allowing us to kind of get global guidance rather than having individual companies or countries that we had to look at the guidances and try to figure out how our method was going to fit within all of those things. So we're very fortunate to have that now, the ICH M10. We do look in the beginning, you want to make sure that you have recovery and that you have the best recovery you can get. As he said, we're trying to look at specificity and accuracy and precision, and trying to get those things dialed in before we move forward into validation when we're in the method development space.
From your point of view, US and most countries over the world are accepting ICH M10 and as their guidance for bioanalytical validation. Is that
Yes,
case
yeah.
from your?
Definitely seems that they're measuring us against the ICH M10,
Nice.
which is great.
That's great. And just as addition, and FDA issued or published a new guidance for a biomarker analysis, which is addition and to the ICH M10, which theoretically doesn't cover the biomarker analysis, although the endogenous analyte section 7.1 is a similar and to the biomarker. So we're at the next stage, probably would deserve more discussion. We have been in the industry for quite some time. We're past colleagues, and we have been discussing this about 2000 and the 2001 guidance, then the 2013 the FDA draft guidance. We talk about it, and then 2018, and we talk about the final FDA. Then we talk about the ICH M10, and each company will compare the gaps and to figure out and compare notes with the CROs. Why we are talking about this method development and the guidance again now? And any thoughts from your experience?
The method is bioanalytical method is critical to ensure the data quality, the accuracy, and this basically is the bottom line to make sure our drug is safe and efficacious for patients. So this is the bottom line. So that's the reason like the regulatory agencies put so much efforts and emphasis like with different regulations. Even like after multiple years, there's still some like improvement or like evolution for how we can make sure our assay is robust, reliable, accurate, and specific to ensure the data quality and accuracy. And also, I think our discussion today is mainly for small molecule. I think even for small molecule, it has been for many years. I think that's maybe it's the longest and oldest modality for drug development. But still, up to now, small molecule drugs are still the most approved drugs. I just did a quick check for the approved drugs of last year. Actually, more than 60% of approved drugs are small molecule drugs. So that shows the importance, even for small molecule, there's still a lot of things we can do for small molecule drug development.
That's a good point, actually. Think about right now, we're accepting ICH M10. So from your perspective, each of us, what was the difference, the M10 comparing to the previous guidance, FDA 2018? I can give example. The matrix spec evaluation is quite different from the previous ones. And what's your thought on it? Jennifer, could you comment on, right?
Yeah, I mean, I think that was the key difference, really. One of the biggest differences was changing from the matrix factor to matrix effects, and the way you did the calculations. It was very specifically described in the guidance and in the white papers that had come out before ICH came out to do the matrix factor experiments. Matrix effects is different, maybe better. And you can still kind of do the matrix vector calculations there with matrix effects, but yeah, it's different, and we just have to adapt, right?
It is simplified. Can we think about what's the reason behind it's getting simplified? Because or the technology advanced, so the matrix fact is less important, or is the instrument is overcoming those? I don't know. What's behind this rationale? Yeah.
Yeah, I think actually it's more important.
Okay.
Yeah, because like the bottom line is like we want to make sure the assay is accurate and reliable, right?
Right.
Actually, the matrix effect is one potential like issue for the assay. Maybe it's like more observed during method development, right? We want to make sure the assay quality. How do we evaluate the matrix effect?
Okay.
Right. So this is, I think, the guidance ICH M10 compared to the previous guidance, you can see there's more emphasis on like we have to test different loss, right? Different replicates at different concentrations, right? That's
Triplicate, right?
right. So
The guidance simplify the procedure,
yeah,
make it easy for everyone to be standardized, and each lab will perform similarly. I mean, we know we have been working at the different places. Each lab, you will do this matrix effect evaluations differently, and some of them will more or less. I think this one just good consistency. I think easier for method transfer. Is that right? Or
No,
any thoughts?
definitely. I think we are also working on complex modalities
I see.
difficult to address drugs, and multiple components often comes into play. And we are going into, like Long mentioned, that 40% of our modalities are non-small molecule. So complexity is coming around there, and also complex drugs are being administered. Because multiple drugs for oncology areas. So it is difficult often until you go into the study to understand the matrix effect. And so it's important that we are doing this in the validation state, method development state.
I see. That's great.
That's what's my observation.
I see.
Yes.
Yeah, it's one of those key experiments you want to do in method development, because you know that that's where it's going to go sideways in method validation if you don't check it, right?
In the old times, some of us just to do a single each lot and just do once. Now it's required how to triplicate, right? That's great. So it's reinforced. I agree. Yeah. Thank you.
Great. Like try to specify in M10, like said about like you need to test like at least 6 individual lots and source.
Okay.
So it's like align like every lab to follow the guidelines.
I see. That's great. And as I said, most of us that has LCMS background. We're talking about small molecules. Actually, at this stage, many of us not only doing small molecules, but also extend our territory into large molecules and modalities. Although that's not the part of the focus of today, but we can reference some of those experiences as well, for sure. We talk about our experience, how to get those experiments done in the correct way. So Long, again, from your perspective, your experience, you have been working in the big pharma and the biotech, and what's the common errors or pitfalls in the lab to prevent us to be compliant with the bioanalytical regulations?
yeah, I think to me maybe the training, a good training is kind of the foundation to make sure people are following the SOPs, following the compliance, especially for GLP, a validated assay, and GLP, like a regulated bioanalysis.
I think, I want to also add is technology has changed a lot
Right.
over the years. Small molecule method development, can you start with high-resolution mass spectrometry so that you make sure that you have your molecule of interest, as well as you make sure that you are separating out the metabolites and interference early on in the method development, and then transferring over to triple quadruple based methods for small molecule. Because a lot of times we get in trouble with bioanalysis when we find metabolites late in clinical studies, and we haven't seen those metabolites as interference in method development, because sometimes animals don't make their metabolites. In vitro systems cannot make the metabolites. So that is important, that we also apply new technology like high-resolution mass spectrometry for small molecule method development. That is changing our field. The technology, like Long said, we are trained more courses, short courses, more specialized courses at many conferences, giving us the opportunity to get trained more and apply these new technology to make sure that we ensure safety and efficacy of the drug.
That's great. You touch upon two good points, and we'll follow that one by one. The first is we're still facing challenges in drug development and clinical trials. How do we handle the metabolite and the complex matrices? How do we bridge into preclinical and with clinical, especially metabolite? When do we start to monitor metabolite, and also what's the stability evaluation requirement? And when we don't have a reference material for some of the potential metabolite, how we manage that, right? You see, can we share some thoughts?
It's very, very hard to quantification for like a metabolite without a reference standard. So we cannot build the calibration curve, and it's hard. Yeah.
Yeah, I mean, I think you can certainly look at VAT conversion of metabolites, maybe using some artificially created, incurred samples, or maybe some in vitro samples that someone could make some metabolites there and look at VAT conversion of some of those labile metabolites. If we don't see them in animal species, you're kind of hit up the side of the head, when you get into the clinic and we have these new metabolites, and it's really up to the sponsor, but it's up to us to advise the sponsor as to what we've seen. We have a lot of experience that we can share with them to help guide them through the process, because a lot of the smaller companies may not have the experience that we have over the years.
That's great.
Yeah, I think this is, I agree, this is a very good point. Sometimes may not everyone has the knowledge or experience, especially for metabolite. Sometimes we see, for example, the glucurolide metabolite.
Correct.
If you don't know about it, then it could cause some interference to your assay. So
Exactly.
even before the method development, we should collect as many information as possible to understand the potential metabolic pathway for the analyte, and then we can design and develop the method accordingly.
I think I want to build on Long's point, and many glucuronides show up later in the clinical stage, and often LC-MS methods cannot separate them out, and in-source fragmentation can happen and cause many issues with the method. So one of the things that Long touched on is early on, before method development, you want to look at the structure if you can. But working in CROs is very difficult to get the structural information.
Right.
So I think working in pharma industry and having the chemistry
The requirement.
we understand it.
Yeah.
So I think the expertise helping small biotechs and other CROs is important. So that's where the technology comes in, and I think JC's point is, without standards we cannot tell how high a metabolite is. But one of the things,
Sometimes you have to do it.
you have to do it.
You have to do it without the reference material.
You have to do it, and sometimes you have the help from other detectors like UV detector and whatnot.
Yeah.
But I also want to highlight that we have new in vitro technology like hepatopac and other system
help to predict
predictive
you to model.
right?
I see. I got you.
So those kind of new technology on the other side, animal models, and then in vitro technology can provide us some information about metabolites early on. But I think key is looking at the structure, where the modifications are.
I see.
And then applying that knowledge for your method development.
I guess from what you said, remind me when we worked together in the past, I see 2 points. First, the communication between preclinical team and the clinical team and also in vivo and in vitro to collect as much information as Long said, and in vivo metabolite profiling, the discovery, and provide prediction and projection what metabolite potential could be formed, especially those phase 2 metabolites. Second, use those in vitro tools to predict the potential metabolites, and also those are internal communication. Then the other one, the pharma, the community between pharma and the CROs, provide information to them during and prior to message development. Actually, I do have experience for one of the glucuronide conjugate. We did not have the reference material. So what we did actually, we used some of the dog or rat study samples. We made some sodium reference material. And if we use a dog plasma, then you evaluate stability back conversion. As you said, chromatograph separation is very important. We change the pH from acidic to basic, then you can separate the parent and the glucuronide.
Yeah.
That's the first. Second, we use the Inca samples as a sodium standard, and for dog and rat, like for human assay, because many these days for many companies, the preclinical, the IND talks, and the clinical, the time is very short.
Right.
And when you start IND enabling, the human assay or first in human study already be in plan. So actually, we use some of the dog plasma to extract and to make a sodium reference material for the metabolite and ship to another lab to evaluate that conversion, the stability, and benchtop and the freeze-all prior to.
In the clinical samples.
Right, yeah. So those are the experience and communication and be innovative and also another thing, metabolite monitoring. It's collaboration between DMPK, bioanalytical, clean farm talks, as well as CMC chemistry and analytical. We should bring them in as the partner. As JC said, if without reference material, we won't be able to do it. We can estimate, but for the accurate quantitation, we need. So we have to inform our chemistry and analytical CMC to prepare those reference standard before too late. Those are good experience.
I just want to add one more point.
Yeah.
I think other guidance you can also look at is metabolite and safety testing.
Yeah.
List guidance.
For sure.
Where that comes into play is that if there are any human-specific metabolites that we want to pay attention, and the guidance documents tells you that if you have any small molecule human-specific metabolite, you have to make sure it's safe.
Right.
And then you have that metabolite tested in animals, because you have 3-month or 9-month tox studies going on. If you don't have the information about the metabolite, then you might be having difficulty to get the marketing approval of the drug. And we have experience with some drugs getting delayed or getting to patients by 1 year because you don't have the metabolite established in preclinical species.
Yeah. Well, and I think the other thing that's important to think about when we're doing LCMS, at least, is just critically looking at those chromatograms, because we can see those metabolites sometimes, and the sponsors aren't going to be able to see them because they're not necessarily looking at the chromatograms. We're just giving them the data. So looking at those chromatograms and seeing, are there other things here that don't seem quite right? They're not in our standard curve. They're not in our QC samples, but they are in those incurred samples, and we see these other peaks. Communicating that back to the sponsor and saying, hey, we might not have this. We might not have the structure. We might not be able to see the structure, but would this be a possible thing that could be a metabolite?
Yeah, I think this is a great point, especially nowadays. We are outsourcing a lot of study, right? As like a sponsor, we may not see the raw data, so we don't typically see, like, if you observe additional peak, if you don't let us know, we will not know about it.
Right.
Exactly. So this, if we like, I think the communication here is really important to
Very key.
right
Yeah.
so we can identify potential issue or potential metabolite. Yeah.
Yeah,
Right.
that's great. Talking about misguidance, actually, the misguidance metabolite profiling can be 2 hours, 3 hours.
Yeah.
If you don't mind, I want to get your thoughts. You see, start from 2008, 2009,
Yeah.
when the guidance first released, and then the high-resolution mass spectrometry became very powerful. Many big pharmas, and they start to, especially DMPK, they start to explore the profiling metabolism using first-in-human samples and MAD in the cold So there are different opinions when you talk to different companies. Some of them wait until late stage to get the human anatomy more definitive. Some of them profile early. So any thoughts on that? Is this still the same or debating?
I think missed guidance just completed a 10-year anniversary or 11th year. So I think we have seen great examples. What I want to highlight is that because we can understand the first-in-man samples, profile those, a lot of companies and a lot of small biotechs can delay their radio label studies because you have the human data early on. So that's an opportunity really created by having the misguidance. And also, one of the other things that Long hit on is that we are doing a lot of global chemistry now. A lot of compounds are synthesized outside of the US, and then we are doing some studies here. So the communication is important, Jan. I think is even starting with the chemistry folks, and then understanding the synthesis pathway, and then what are the metabolites. Sorry. Yeah, go ahead.
Yeah. I mean, I would say I've seen several examples, even in the past few years, of people waiting too long to do their metabolite studies. They're not looking at their first in humans necessarily. They are going back and digging out old samples and reanalyzing them, 4 or 5 years later, and having to try to come up with stability for some of their metabolites, or repeating a study to be able to do
some of the talks.
to read, well, or the human studies, to be able to get some of that metabolite data, to be able to move their program forward. And so making sure that we're advising people, this is something that we see. You should really look into this because it's important for you to catch this early, so we're not having to play catch up on the back end.
We use chip quadrupole mass spectrometry a lot for small molecule bioanalysis. So some of metabolite may co-elute with N-light, and we cannot see the peak shape change. But as a new technology, high resolution mass spec. So if you to go through search the M over Z in the peak, so you probably see the metabolites in co-elute with that peak. Then talk with sponsors, inform them it maybe have something there, we can dig into that.
Yeah.
We should do some method development that change the different chromatographic conditions and trying to identify some potential metabolites and which were not seen in other conditions. I guess as a summary, during early method development for clinical, the communication very important from DMPK, get the knowledge and to guide our method development, what assays and what analytes we should go after. And as said, metabolites, we can talk about hours and days. Maybe we pause that and revisit that. Let's talk about the method development, automation. What's your experience view, automation as well as the new trendy word AI? Any thoughts on the automation and AI from your experience? And Jennifer, could you?
We use automation some. We are in the regulated space still shying away from AI, I think, for the most part. It's hard to know where the data is being stored, and trying to get all of that information with confidentiality and for our sponsors. I think my guess would be that most CROs in the regulated space are shying away from AI pretty heavily, because it's too hard to control, and make sure that the confidentiality is there for our sponsors. And I think that's the biggest hurdle that I see, from our perspective. I don't know if there's others that see other hurdles.
Thank you.
We also try to use automation like a TCAN, like Hamilton, to do sample preparation and extraction. So for method development, of course, it's early stage, early stage you need to develop method. So it's scripts, programs, it's from scratch. So I need an experienced scientist to program that. And for AI, definitely it's a different topic, but it's really hard. I believe in the near future, definitely we probably move to that direction.
For sure.
That's good.
One of the things going back to automation, we have to also look at it. It also allows the scientists to work safely.
Okay.
because some of the modalities, I know we are not touching on the large molecules.
Yeah.
Some of the modalities, some of the matrix could be difficult to handle, and safety-wise, automation gives us that option, like the Hamilton system. If you look at it, sample analysis from non-human primates in human samples. So it is
They're all under the hood,
under the hood.
enclosure, right, right. Those are the current standard.
Right.
Yeah.
So it's important to have that in mind. But one of the things that you have to keep in mind is that return on investment for pharmaceutical companies and also CROs, because maintaining Hamilton system, if you don't have enough sample throughput,
see.
then manual approach might be better in certain environment.
Where you can do step by step,
by step.
or I know if you do a programmed
Yep.
screening, you say I'll screen 10 columns and 12 mobile faces overnight, but then it may take time to review the data, and that can be pitfall. Then we have to have a good balance and
Right.
depending on the need. That's Long. What's your experience? And
Yeah, I think for automation, maybe it's more like for the
Production?
production,
I
right?
see.
Because for method development, different compounds, sometimes it's hard to apply a same automation or same screening system for everything, right? You still have to do some manual, like method optimization later. And also, I think it also depends on the size of the company, right?
Okay.
If you do like 100 compounds
Right.
per year versus you only do like 10 compounds per year, right? So the automation maybe better for those you need a high productivity, right? Those type of.
I see. So you're saying we
yeah.
have a more flexible strategies at the different companies at a different time, different stages, and sometimes we'll apply the automated to screen to get the baseline information. Sometimes if we understand the modality or the therapeutic programs quite well, the compounds are quite similar, then we'll just take some existing method to modify instead of starting from scratch. To be more
And trying to get all the information you can about a compound ahead of starting method development. If there's no knowledge out there at all, then a more extensive screening process is probably more important. But if somebody else has already developed a method for it, why not start there and maybe tweak it a little bit so that it works in your lab, works with your system, that kind of stuff.
Another topic, and which could be a little bit controversial, and but we want to touch upon, and really we're in an ever-changing world, and the global geopolitical situation, and it's different from years ago, depending on, regardless of our political point of view, we're facing some geopolitical situations in terms of the regulatory guidance or changes. For example, FDA may have some structural changes, and as I'm aware of, and some of the inspection may have some delays, and we're not sure if any changes in FDA priority and their structure would affect our bioanalytical method development. Can we foresee and some potential issues to prepare? For example, if any changes in FDA, would that possibly delay any new guidance or modification? And what can we do? What we can do to help ourselves in this situation? Any thoughts on?
I think communicating, trying to make sure that we're all holding ourselves to the same high standards, maybe bringing in innovations ourselves, and trying to tighten those standards on ourselves will help the FDA, whatever stage they're in, whatever form they have, any of that will help them feel more confident that we are holding ourselves to high standards, and not letting things slip, even if they can't be inspecting us as often, or whatever happens. Who knows what's going to happen? Yeah.
I think the communication with FDA or agency early on
Right.
is important. So if you can get that face-to-face meeting or review or whatever form you want to have, so that FDA also has a lot of experienced scientists who have trained in pharma or CROs and working there. So it is important that we have that establish communication. But in a globalized pharmaceutical discovery and development, we have to be open to looking at other opportunities in outside of the U.S. For reasons that if we have resources, difficulty developing assays here, and then we have multiple biotech companies. They have limited resources. They may not have any lab space and want to get those experiments done timely and the scientifically sound experiments. So I think it's important to make sure that we have the opportunity to function in a globalized pharmaceutical discovery and development and in bioanalysis also. We also have European guidances. We have other guidances also helping us to make sure that we achieve this.
That's a good point. And you're saying, although the global US, EU, and Asia are following ICH M10, but as we're aware of, actually, European bioanalytical community somewhat following slightly different, not say different guidance, they have some different approaches. For example, the non-fully validated assay, they have the scientific validation concept. And so what are we foresee any potential in US if the validation standard can be relaxed, or are we have any concern, any potential quality control issues and for validation US? Then another, on the same point, are we foreseeing more clinical trial studies move to other countries, into Europe or Asia, to avoid any uncertainties in the current political situation, if we can say that? Any thoughts on that?
I think now like ICH M10 is a global
Right,
harmonized.
right.
So in the short term, so these guidelines still like effective, and in short term, like we still need to maintain the strict like method bioanalytical method validation requirements. Maybe in the long term, like there are some new technologies like real-time PK monitoring. So those things in the new like guidelines may be delayed. But anyway, farmers and biotechs, I think the best is maintain strict like bioanalytical validation requirements to avoid potential regulatory compliance issue.
Yeah, I think one other technology that we also want to think about is remote sample collection, because difficulty of doing clinical studies with patients and doing studies in larger population, the remote sample collection and watching somebody providing sample on a video or something, make sure that they're giving the right sample. And so that you can globalize some of these clinical trials. Difficult to find patients for certain disease is important because they are very complex. So if you're looking at CROs, sometimes you're getting samples from 5 different parts of the US because patients are spread out, and we are going with patient treatment early on to provide that opportunity to benefit from new modality and new drugs in this case. So we also need to look at those technologies in a
Technology, also globalization.
globalization.
Right. And that's good. And Loam, you see now there's concerns for the rising drug prices in US. And are we seeing, or what's your point of view, any pressure for the pharmaceutical to cut cost and cut the R&D budget and also outsourcing to CROs? Any thoughts you experience?
Yeah, I think cut cost saving, like more cost effective is always the path to go. I think a lot of companies are under pressure to have a reduced R&D cost, right? So in this case, for bioanalytical, I think we really need to think about it. Is the assays really need to have, or sometimes it's just nice to have, right? So maybe I'll go back to the metabolite again.
Yeah,
right?
right.
Sometimes like maybe it's good to analyze all the major metabolites, but sometimes we also need to think about if we already have a missed coverage, of those metabolites. If it's an inactive metabolite, do we still need to monitor that, right? So if we can reduce the work for the metabolite assay, something can be more cost effective.
That's a good point. Actually, now we're talking about the drug price and cutting costs, and also we're talking about earlier about comprehensive method development, and Raghu brought a good point to be fit for purpose. And so with the budget cut, what's your thought on we do a more focused, more effective bioanalytical method development, just as Long said, instead of a comprehensive, right? What's your thoughts on that?
No, I think this focused method development is an opportunity
Okay.
to make sure that, we just, we're getting the information we needed, but also we have to make sure that method is scientifically sound. So I think I'm going back to my point about if there's a metabolite interfering, if there's a matrix effect, or if there's a biomarker that you are interested in, can we get those information?
Or paper exercise, all the information,
Exactly.
virtual correlation and prediction,
Yep.
get all the ways. And
I think, I want to go back, Janice,
Yeah.
also software development.
exactly.
So, like ACD software, and you put in the structure.
So are you merging to AI? You're talking about the AI. It has a lot of AI.
AI will be a different discussion, but this is software tools. A lot of mass spec vendors
Okay.
are providing now.
Yeah.
That you could put in a structure, and then you can get theoretical metabolites, theoretical interference, theoretical method. So those are the advantage that we have today with new technology. So I think in a globalized drug discovery and development, I think we need to take advantage of those because we can do that often remotely. But remember that in a CRO environment, sometimes it's difficult to get a structure.
And Jennifer, if we still do it on the cost topic, what's your thoughts on generic and biosimilar? And are we going to see another push for generic and biosimilar for the low cost? And what the pharmaceutical is going to face, or any thoughts on that?
I don't know if I have a lot to say about that, or that's more the pharma side or the biotech side. Certainly, from a consumer perspective, the generics are certainly helpful for us to be able to get the drugs out to the people who need them, and I think that is the goal of the larger industry for sure. To get everything out there so that everybody has access to it, and not just people who can afford to spend millions of dollars on healthcare. So being able to support those things is certainly something that we do, right? So we're looking at the BE studies, the relative bioavailability studies, to make sure that we can support those and we can follow the regulations and follow everything that's in ICH M10, to allow us to have the FDA accept those studies when they come out of our labs.
That's great. I guess, might there be a faster review for new drugs to reduce the cost, still on the cost? And if there's a reduce the cycle for drug approval from FDA for regulatory, and will this affect our method development? Would that benefit our CROs and pharma? Any thoughts, GC?
I think of course if they want to accelerate the approvals, so of course less time for method development, right? And they need to analyze more samples, provide a high quality PK, PD, and BE data, right? And sometimes they probably need doing method development, method validation, and sample analysis. At the same time, they prepare the regulatory submissions. So of course, so that's the trend. They want get a drug approval as soon as possible to saving lives. Yeah.
I think one other point I want to make is bringing a drug from discovery into the hands of patients takes millions of dollars, sometimes billions of dollars. So if you're looking at generics and if you're looking at other options, one of the options I really like is where universities, local institutes get into drug discovery and then getting the drug to the point that you can take it further with the pharma partner or CRO partner in this case.
Just like a biotech and develop this,
In the
give
university,
to the big pharma, right? It's similar model.
Exactly, and the
The
university
intellectual property
Yeah.
and the knowledge.
university has a lot of opportunities
point.
with chemists, and a lot of universities, I don't want to mention names, but they do this today. And it is a way to reduce the cost and bring the drug faster possibly. And so it is important that, to your point, that how can we reduce the cost? Going generic is another one, but pharma is already
Reduce the R&D cost,
R&D
right,
cost.
by incorporating the university and academic resources. I think that's a good point. And
It's
great.
an opportunity.
Maybe the last topic around the cost. You see, there's a push. There could be a push for U.S. domestic drug manufacturers, right? Likely that will increase demand for U.S.-based CROs, and instead of relying on the imported standards, cost reagent, and this, which could have a higher cost. Any thoughts on this? And back to U.S. manufacturers, CROs?
Yeah, I mean, I think we're certainly keeping an eye on this, right? So this is something that we've been watching for 3, 4, 6 months. Looking to see, are there reagents? Are there tubes and pipette tips and 96-volt
Supply
plates,
chain,
any of the
supply
other
chain,
units,
all
or are there
these.
any supply chain issues with these things that may not even come directly from the consumable that we're using, but from the upstream materials that have to be made into those consumables, and keeping an eye on that and making sure that we can still keep buying things. I mean, we all learned from COVID, right? That we stockpiled all sorts of things, all sorts of places to be able to keep going in those times. And so, I think the nice thing is that I'm not sure if that was a nice thing that we had that experience, but that experience has taught us something. We came out a little bit smarter out of that, and it taught us how to keep going in times that were pretty tough back then. So.
Anything? Kind of thoughts?
Just want to add, I think COVID taught us, how to work pipette tips. We were not able to get it. We learned how to wash them and reuse them. But today, I think when we have access to it, often we dispose it, and we come back to this question again. Can we get these pipette tips in the US? Can we have these, available for us for next 6 months?
Right.
So really, I think I like what we learned as a drug discovery and development environment. But going back, we learned different ways too with COVID-19.
Do you see any? I agree with you from your perspective.
Yeah, so
here.
from pandemic, it's drug like a supply chain, consumable supply chain issue. So like the lab, if you don't have consumables, how do you run experiment, right? So you have to make sure we have easy access to those consumables. Make sure our experiment can conduct and finish the sample analysis.
That's great. Lo?
Yeah, I think sometimes we have to find some different way or smart way or alternative way to work on it. Maybe as the pandemic as an example, I think during that time, not only the suppliers like some biological matrices like the monkey,
Yeah.
CSF, so we cannot purchase it, nowhere to purchase it, right? So we kind of switch to use surrogate matrix, like surrogate artificial CSF to develop assay, to validate assay. And I think this works very well. Actually, it saves us huge cost, and also we don't have any supply issue with this. Yeah,
that's great. So keep perspective. Tomorrow will be beautiful, right? I guess as a recap, today we discussed why we need to do method development and the validation, how important it is, our shared experience and in the actual work, and how we conduct the method development and develop automation, and also this metabolite guidance, and also new technology. And also we touch upon, we're all scientists, we touch upon some area we were not familiar with, and the political and influence to the pharmaceutical, to our work. That's great. I think I learned a lot, and hopefully we still have some unfinished business like AI and some globalization. Hopefully we can come back to revisit those topics maybe in a few months. How's that?
Yeah.
Thank you, everyone.
Yeah. Thank you.
Thank you.